Okay, so as a summary, so first I discussed how is recursive production of cells possible in the midst of diversity and fluctuations. So in this model, there are several chemicals, and these chemicals mutually catalyze each other and produce the cell. And so when the number of molecules within there increases, then it divides into two, and this repeats. And so the question is that how this recursive production is possible. And what we found here is that this is possible at some kind of critical point of this reaction network dynamics. And there, so a hierarchy of catalytic reactions, so cascade catalytic reactions are formed. And then at this point, we found some kind of universal statistics about the abundances of chemicals, that is Zipf's law on the chemical abundances. And this is so confirmed by the experiment of gene expression data. And then also we checked this, the distributed the fluctuations by cells, and so each chemical component differs by cells. But this fluctuation is found to obey the log normal distribution. And again, this is checked, confirmed experimentally. And then, so this log normal distribution has a very large fluctuations. So we discussed the possible relevance of such large fluctuations. And first, we did some experiment to compare with this phenotypic fluctuation and evolution speed. And we found that this phenotypic fluctuation variance is proportional to evolution speed. And that is explained through the fluctuation dissipation theorem. So by extending fluctuation dissipation theorem in physics. But that means, so this phenotypic fluctuation is a fluctuation of a clone. So usually in population genetics, they discuss the evolution speed in relationship with the variance of genes. So by changing genes, so by the mutant, the phenotype change. So this phenotypic change is in accordance with the genetic change. So now this, and in this case, so this genetic variance is proportional to the evolution speed. So if these two are so both true, that means there is some kind of correlation between the phenotypic fluctuation of clone and genetic variance. And so we so argue some theoretical explanation to show that these two are so proportional. And then we check that this is true by using the simple toy chemical reaction network cell model. So then another question is that about this large fluctuation of molecules. So the question is that if this is true for all molecules in the cell. So then, so we discussed that some specific molecules, so the fluctuations are suppressed. And so to be specific, we consider some kind of mutual catalytic reaction process, and two types of molecules catalyze each other. And then in this case, those molecules that are minority in number play the role of heredity carrier. That means this is very well preserved and controls the behavior of the cell. Mm-hmm. So there is kind of symmetry breaking between these two molecules. So one group starts to control the other group, and this group is a minority in the cell. So that means the minority molecule in the cell or the proto cell becomes the gene carrier, genetic carrier, and probably that's the origin of the genetic information, and so why DNA looks so important in the cell. And to confirm this statement, we did some experiment using so two types of molecules, and again DNA and enzyme. And then we found that this experiment, when the number of DNA molecules is small, then so this cell can can continue reproduction. But when the number of DNA molecules in each tube is 100, then this reproduction activity will be lost in a few generations. And at the last part, at the last part, so we discussed about the multicellular property. So considering a cell within some chemical reaction network, and then as the cell the divide, then there are many cells, and these cells interact with each other. And in this kind of interaction model, we found that there appears generally some kind of cell differentiation process, and in this case, robust development under fluctuations is possible, and also irreversibility of cell differentiation from stem cell to other determined cells is generally observed. And this mechanism is that phenotypic differentiation due to instability of homogeneous states, cell homogeneous states. And then this forms a discrete attracting states by cell-cell interactions. And so at the last point, there is a remark that in biology, in contrast to just dynamical system studies, selection of dynamical systems rule itself is an important issue. And in the study, we discussed about genotype and phenotypic relationship that suggests that dynamical systems rule, that is a gene or parameter, somehow assimilates the dynamics itself, so phenotypic fluctuations. And we confirmed this statement first in the model of this simple toy cell model, that abundance distribution, that is zip flow, is later embedded into the network structure through evolution. And in this case, finally, it leads to scale-free network. And the second, we discussed how these genetic fluctuations and phenotypic fluctuations are correlated through the evolution. And finally, this time I could not talk, but in the theory of sympatric speciation, we previously proposed a theory that first there appears bifurcation phenotypes induced by interaction, and this differentiation phenotype is later assimilated or fixed to genes, and that leads to stable speciation process.